Hi, everybody. We're going to get started, so if you can get a seat. People in the back, you might see better in the front, so plenty of seats. Today, again, we have a great lineup, not counting me. And first up is Edoardo Marcora, our colleague from Uptown at Mount Sinai. And so he's going to give us a short talk on from human genetics to causal biology in Alzheimer's, one, of course, of the disease that many of us study. And so it was a pleasure to hear Edoardo speak, so looking forward to it. Edoardo?
All right, let me make sure I'm pressing the right buttons. Yeah, so this is sort of, I shortened the title. I thought that was too big, too large, but this is really exemplify what we are trying to do at the Loci Center, directed by Alison, which is to translate the human genetic findings into an understanding of the causal biology of disease to then drive drug discovery efforts, because we really believe that understanding biology is key to develop effective therapeutics and hopefully preventative strategies as well. So this is sort of a schematic of how we proceed in trying to achieve this goal. So, and the talk today will be mainly dealing with this first blue part here, which is the integration of both human genetics and human genomics for the identification of what the functional causal elements are for disease, the variants, the regulatory elements, the genes, and the pathways that we believe may be affecting risk for Alzheimer. We follow this up. I won't talk about much of this today, both in vitro and in vivo research done with human or engineered cells as well as mice, and following this with early drug discovery efforts in collaboration with consortia or pharma companies themselves, as I'm coming back actually from pharma to academia a few years back, and I'm touch on this. So I would say our assumption trying to tackle the complex genetics of sporadic late onset Alzheimer's is sort of based on what we have learned from the monogenic forms, the autosomal dominant early onset forms of Alzheimer that are caused by mutations, coding mutation in 3 genes, which, as everybody in this audience knows, are sort of participate in a common shared biochemical pathway called the aminogenic processing of amino precursor protein. From these findings, of course, the amino cascade hypothesis came about, and that led to discovery efforts, unfortunately, largely failing, at least up to this point. But for the late onset version, we have a much more complex genetics. These are polygenic disease, and we have GWAS, fortunately, that can kind of give us a clue as to where in the genome the association signals are. And the challenge, which I'll be talking mainly about today, is to translate these findings into, first of all, a catalog of genes that, again, we thought are causal to the disease, and also then trying to understand how these genes interact into molecular interaction networks or cellular processes, which then will be prioritized as causal to disease. And of course, drugs and therapies will have to be designed to affect these processes and genes. So this is, sort of, I think, a rather misleading way that geneticists label Manhattan plots. For those of you who doesn't know, these are basically the chromosomes, and this is the association signal, in this case for Alzheimer's disease. This is the IGAP GWAS, one of, not the latest, but, rather large GWAS dating back to 2013. And, you see this peak of associations, and I guess mainly for mnemonic use, the geneticists like to label them with gene names, normally the gene that's closest to the center of the associated region. And that's really misleading, because most of the biologists don't know this, and they think that the genes are actually mapped and understood, and that's not the case, as I'll show you in next slide. So just taking, for example, one of these loci, the self-1 locus, and zooming in, you can clearly see that there's not just one gene in there, and there are several genes just underlying the region of association. And the problem actually is that most of these associated variants are non-coding, so they don't point directly to a gene. But as we learn from other GWAS studies, other diseases, they are mainly regulatory in function. So they are they affect the expression of genes and could be expression of multiple genes, some of which may not even need to be under the associated region. They may be megabases away from the region of association because of chromatin looping, et cetera, which I'll be showing you in a moment. So, so this is the one example and that we tackled, and I won't talk about that, but just briefly. But this is basically the sort of what is generally known, but we have sort of, when using this, this sort of model to try to understand Alzheimer's disease genetics, is that most of the GWAS SNPs are actually tagging haplotypes, which then affect the function of regulatory elements, for example, enhancers, which then are brought through, for example, chromatin looping, et cetera, to the proximity of promoters, which then to affect their regulatory function onto the expression of a gene or multiple genes, and that the risk alleles are basically affecting the function, for example, the binding of transcription factors to the enhancer, thus affecting the expression of the target genes for that specific enhancer. Sort of, it's understandable there's sort of a bit of unease in kind of moving away from the gene as the unit of genetics, which is definitely mainly the case for Mendelian disorders, away from the gene and kind of start to think about regulatory elements as the actual unit of burden for common variants affecting risk for disease, because, you know, unlike genes, which are pretty well annotated and at least in boundaries, they're pretty well conserved across the cells if you take splicing away, but the regulatory elements are highly cell type dependent, and not only they're highly cell type dependent, they're also dependent on the state that the cells find itself in. So it's really hard to map and to really understand among the many cell types, and I mean, I'm just showing just brain cell types, and they don't have to be brain cell types either. Among the many cell types in the body, what's the causal, the most likely causal cell type, and allowing us to sort of use this information derived from the cell type to sort of map the genes and the targets for the risk variants. So in order, you know, sort of there is already some ideas as to what the cell types or cell state may be most likely the causal ones for Alzheimer, coming from an early paper from actually Phil Jager's lab, the next speaker, who introduced me, showing, in this case, looking at, I think this paper was mainly about trying to understand the contribution of the adaptive versus the innate immune system in various diseases by looking at the polarization of cis-QTL effects and risk variants for these diseases in either monocytes, as sort of the exemplary cell type for the innate immune system, or T cells for the adaptive immune system. And among the diseases that were enriched for myeloid signal and impoverished for T cell signals, Alzheimer was actually at the very top here, showing again an enrichment for monocyte colocalized effects of in monocytes. We sort of extended this, well, I guess turned this approach on the side, and instead of looking at a few cell types, two cell types across many diseases, we look at pretty much all the cell and tissue types we could put our hands on across one disease, in this case Alzheimer's disease. We also use schizophrenia just as a control, and found that, you know, through a different approach, which is looking for enrichment of AD risk alleles in epigenomic annotation that are cell type specific, and found out that beside liver, there were no actually signal in brain tissues, and much of the enrichment was observed in blood cell types of the myeloid and beta lymphoid cell lineage, which sort of goes with what Phil has shown before, but sort of extends this to across cell types, and you know, this was quite illuminating to us. And this is the year later was confirmed by this other paper from Alkas Price, who basically combined Phil's and our approach, and they look at all cell types in all diseases they could put their hands on. And for Alzheimer, they also found that both by looking at epigenomic annotation like I did, or you know, gene expression enrichment, found that myeloid cells were the cell types enriched for Alzheimer's disease irritability. So this sort of told us that looking at myeloid cells may be at least a good place to start. And in this paper we published a couple of years ago in Nature Neuroscience, we used the integration of both the GWAS information in terms of summary statistics and expression QTLs derived from myeloid cell types like monocytes, and triangulate these two layers of information to sort of arrive at the prioritization of candidate risk genes for AD that are in myeloid cells. And in using this approach, and going back to the self-one locus, which I've been telling you about at the beginning, we were able to, not only based on this evidence, but other sort of confirmatory and convergent evidence, we were able to prioritize P1 within this locus. And I'm not going to talk about this paper. It's published. You guys can look it up if you're interested. I just wanted to mention it here because P1 is actually encoding a transcription factor called Pu.1, which is well known to immunologists, as it is the major sort of myeloid cell lineage determining transcription factors for myeloid cells. And we not only found that modulation of the expression of this transcription factor is associated with modulation of AD susceptibility, but that the target areas of the genome, the binding sites for Pu1 in the genome, are also enriched for AD risk alleles, pointing to perhaps sort of not only P1 itself, but its cystrom as being enriched for genes that play a role in Alzheimer's disease, which we also then validated. And this is interesting because P1, together with MEF2C, which also interestingly is another transcription factor that resides within another AD risk locus from the same GWAS that I showed you before, act together in microglia to basically set up and establish the pattern of enhancer activity in microglia, and which are the myeloid cells of the brain. And this really made us starting to think of enhancers as being the unit of investigation that we wanted to concentrate on. And this is sort of the approach that we sort of extending from the previous one that we took in this paper that's been recently, a couple of months ago, 3 months ago now, put up on bioRxiv in review now, which the talk would be mainly about. So we, at the same time that we published this paper, Christopher Glass came up with a great resource, which is basically both transcriptomic and epigenomic marks from microglia isolated from human brain. And we could use this resource to sort of extract the location of regulatory elements in microglia as well as in other myeloid cells, and divide these regulatory elements in 4 categories: active enhancers, active promoters, prime enhancer, and prime promoters. And as you can see, by performing the same sort of heritability enrichment protocol that I showed you before, we found, well, replicated what we saw in the original paper, but we validated this observation in peripheral myeloid cells with myeloid cells from the brain, i.e. microglia, and again, saw actually this interesting enrichment for AD risk alleles that is significant only within active enhancers. We then went in to look at what transcription factors that are expressed in this cell type may be burdened by AD risk alleles, and within this enhancer, we took the ATAC data and sort of labeled them by the presence of the binding, the consensus motif for the binding site of various transcription factors, and look again for enrichment of AD risk allele within this sort of in silico ChIP-seq, and found that Pu1, as we've shown before, is enriched for its cystrom, is enriched across the cell types, and there is some, some other transcription factors together with Pu1 that appear to be significantly burdened by AD risk alleles in the different cell types, but Pu1 is across the board. So again, we decided now that we established that AD risk heritability is sort of enriched in this type of regulatory elements, we decided to take the regulatory element as the unit of investigation and sort of extended the initial approach to integrate this information. So it's a bit complicated. Actually, we took 2 analytical approaches, 2 parallel approaches, and also look for convergent evidence for these 2 parallel approaches. So the one that I showed you before only took advantage of a variant having either an expression effect on a gene or a risk effect on the disease, and kind of triangulating, integrating this two layer information, we were able, through SMR and other methods, prioritize genes as being causal in the causal chain to disease. But now if we take the enhancer as an added layer of information, we proceeded in two ways. The upper way is to, first of all, filter variants for being located within an active enhancers in the different myeloid cell types. And in one, we use 3 layers of information. One is the interaction through promoter capture IC of the enhancer with its target genes, and the presence of also QTL effects for these genes that are linked to the enhancer through the chromatin interaction information, and then use the risk effects to, again, prioritize the genes that are target to the enhancer in the way they affect disease. In another approach, we sort of did in silico, QTL-based mapping of enhancer to its target genes by taking not only the QTL and the GWAS information, but overlaying that and integrating that with Easton mark QTLs as a way of measuring the effect of variants on the activity of an enhancer, and use sort of two rounds of SMR. Basically, the first round to establish which genes the activity of an enhancer is linked to through Mendelian randomization, and then from the gene expression under this enhancer's effect to the disease risk again. So what time is it? How much time do I have? 10 minutes? Okay, I'll skip a couple of slides. I'll just go to the chase here. So this is showing for just 2 loci. We did this genome-wide. We didn't just focus on one locus like we did in the original paper. We did this across the genome in the genome-wide significant loci and some of the subthreshold loci. And we are showing here, for example, the BIN1 locus. So here we have the GWAS signal, EQTL signal for monocytes or macrophages. Unfortunately, we don't have much QTL data sets yet available in microglia, and when those will be available, it would be great to confirm these findings. But because of what we observe in terms of enrichment of heritability, we believe that using peripheral blood myeloid cells are a good either proxy for microglia, or they could also be the causal cell types. We actually don't know at this point whether central and peripheral or peripheral microglia and myeloid cells are the causal cells. So as I said, these are the GWAS, the QTL in monocytes and macrophages. These are the gene locations, and then these are the in red the active enhancers that are contain SNP associated with AD risk, and then for the approach number 1, the sort of the promoter capture IC interactions between gene and enhancers, and these are sort of the SMR-derived interaction between enhancer and gene through the 2 rounds of SMR that I described above. As you can see, they actually, not always, but more often than not, they actually overlap, and the same thing repeated for macrophages. Unfortunately, macrophages, we don't have availability of chromatin QTL, so we could just do this in monocytes. But this is showing, for example, that these 2 enhancers, which we call AD risk enhancers, affect the expression of BIN1 in this locus. And this is another example of the TP53 IMP1 locus, which we call this way because this is the gene that within this locus we prioritize through these 2 approaches that I described. It is actually quite interesting in regards to the BIN1 because 2 months after we deposited our paper in bioRxiv, Christopher Glass deposited this paper and used sort of a different approach, but in a way also related. He actually generated a beautiful resource looking at both chromatin marks and ATAC-seq and plaque-seq, which is also probing chromatin interactions in the 3 major brain cell types derived from iPSCs, and we have the iPSC, microglia, neurons, and astrocytes, And actually, so these are actually primary cells. And in the BIN1 locus, he sort of, he didn't use much of the genetics in the statistical analysis, but he used the enhancer mapping and the chromatin interaction to sort of link a region of where SNPs are present to a candidate target gene. And in the case of BIN1, he kind of arrived to the same enhancer that we identified, and he actually proceeded to validate this in, actually this was done in iPSCs, where he basically deleted through CRISPR, deleted these enhancers, and discovered that this enhancer is specifically acting on BIN1 in microglia. As you can see, the effect of knocking this enhancer down on the expression both at mRNA or protein level is only present in microglia. We also had in vitro validation from some of our finding. It wasn't on the BIN1 locus. We actually focused on the MS4A locus because this is related to other projects that we have in the lab. And making a long story short, yes, we discovered that within this locus we have this CTCF binding site. Actually, there are two binding sites. One here that kind of overlaps with the GWAS and enhancers in this, under this GWAS region of association. There's this loop that's maintained by two CTCF binding sites shown here. And within this, one of the two CTCF binding sites, this one, we have the SNP, that's the top SNP in this locus in terms of AD risk modulation. That's actually changing this C in the consensus binding site for CTCF to a T. And this is predicted to really have a major disrupting effect on the binding of CTCF, and this is actually observed in the binding QTL data sets that we have from both lymphoblastoid cell lines and macrophages. And given this, predicted and also measured, at least in some myeloid cells, impact on the CTCF binding, this SNP is also predicted to disrupt this looping. So this is our sort of cartoon, our proposed mechanism by which, in the presence of the C allele, we have this looping occurring. The MS4A gene, for which we have an EQTL effect for this SNP, is under the influence of a not highly active regulatory element. As you can see within this loop, chromatin marks are highly repressed compared to the outside of the loop. And in general, CTCF loops mark regions of active and inactive chromatin. And so we think that in the presence of the loop, the MS4A expression is regulated by a primed enhancers that we map here, but not very highly active enhancer. And when the loop is disrupted, the MS4A gene goes under the influence of a stronger regulatory element, which then causes this EQTL effect. We were able to, at least preliminary validation using iPSC-derived microglia from cells that, from carriers of either the C or the T allele. Actually, from heterozygote carriers, we could do basically allele-specific ataxic and show that in the presence of the T allele, which is, given this matrix here, is predicted to disrupt the binding of CTCF at that site, we have much less coverage of ataxic reads at this site compared to the carrier, to the C allele. So this is our at least preliminary validation for the MS4A predicted functional variant and target gene. So here, just to recap, this is sort of showing, since as I told you, we did this across the genome, showing the genes that we prioritize as the most likely candidate. These genes within this loci identified by either one or the other approach, but most of them actually, the two approaches converges on the same causal gene. And in the color coding indicates genes where lower gene expression in blue is linked to increased AD risk, or higher gene expression in red is linked to increased AD risk. And this is also an important piece of information. Start thinking about therapeutic hypothesis and how to go after some of these genes, perhaps as drug targets for Alzheimer's disease. We do have indeed some efforts around both MS4A's locus and the SP1 locus. And so we are targeting those genes with trying to develop assay to screen for small molecules affecting the activity or the expression of these genes. But an important thing, I think, when thinking about polygenic disorders is not to focus too much on the single genes, but sort of the network and the pathways that would need to be drugged, not so much at the single gene level, but at the level of hubs and networks and pathways. And this could either be coming from targeting something like PU1 that drives large parts of the network, or, for example, polypharmacology, sort of targeting the appropriate hubs. But a lot of work needs to be done to arrive to this. But I wanted to mention this because what was really apparent when we started looking at the genes that we prioritize is that most of these genes actually are coalescing around the endolysosomal pathway, which has been implicated in AD before for Ralph Nixon and others, but in neurons. So our study, I think, suggests that endolysosomal dysfunction in myeloid cells may be driving the modulation of AD susceptibility. And these are the genes that, some of which you have seen in that Manhattan plot. The BIN1 and RIN3, actually, these interact with each other, and they are RAB5 effector. RABP1, which is also RAB5 effector. This is, of course, an important protein in early endosome trafficking. CD2AP and ZIX, which modulate acting filament dynamics, propelling endosome around the cell. APBB3, which is also located in early endosome, which is an APP binding protein. AP4E1, AP4M1, which are also in a complex that mediate retrograde transport, actually bidirectional transport from early endosome and the Trans-Golgi network, and SPP. I did something I wasn't supposed to do. SPP2LA, which is actually a presenilin 2-like protein that localizes, it's a protease that localizes to late endosomes and lysosomes. And with that, I'm half an hour, yay. I want to thank mainly Gloria Novikova, which is a second-year graduate student. She's fantastic, very determined, capable, motivated graduate student. Hopefully I'll be able to keep her for a couple of years, but that'll be hard because she's a machine. And so she did almost all of the bioinformatic analysis. Julia was provided the IPSL work, Manav, and help with the SMR analysis. Kay, who is our sort of trade union with the cardiovascular genetics community, which provided us with some of the data sets that we've been using in this analysis. And of course, Alison, the director of the center, with which none of this would be funded or possible. So that's it, and I'll take questions.
Thanks. You talked about the enrichment of the Alzheimer's GWAS, RIS, loci in 3 different cell types from the same lineage. Can you talk about the prospect of more conclusive statements about specific cell types, whether it's microglia or macrophages?
We don't see any.
But not from your study, but just is a specific cell type conclusion, is that going to come from genetics, or is that going to be something completely different than that I don't think about?
Of course, people think of microglia as the most likely myeloid cell to be the culprit, but that's just an assumption, or I think it's a valid assumption because it's the myeloid cells in the brain, and the brain is the organ that's mostly affected. But there's other myeloid cells in the brain that are not parenchymal, like perivascular macrophages and other myeloid cells in the brain. And there are also myeloid cells in periphery, which may modulate risk for vascular inflammation, which then can reverberate in the brain as well, or metabolic. TREM2, of course, is one of the genes that have really propelled this idea of macroglia in Alzheimer's disease, but that is also not a macroglia-specific gene. The only thing that I would say points to microglia in the genetics or coming from the genetics is this PU1 and MEF2C combo, which I talked to you about. were from experiments by Mira Merritt and others in mice, appears to be the combination that's responsible for driving a macroglia-specific pattern of expression and epigenome activity. So if you take those two signals together, then that's really the only thing from genetics that to me points to macroglia more strongly than other myeloid cells, but and that's in line with macroglia being in the more proximal to the site of damage, but we'll have to see, and perhaps just, we don't know until we do phase 3 trials, and if we target macroglia specifically and we have an effect, then we know it's macroglia, but it's probably very difficult given that most of these genes are not macroglia specific. So.
In cases where you find cancer regulatory elements or hitting on a gene, we also have coding variants, so we have kind of a cross-validation TREM2, for example. Do you have any?
Yeah, no, we haven't found any. For TREM2, I mean, there are other genes in the TREM2 locus, like TREM1 and TREM2, which are maybe affected by common variant regulatory mechanism, but for TREM2 we haven't found a convergent like gene expression regulation mechanism and a rare coding variant mechanism. So at least for TREM2, so far the main effect seems to be coming from the rare variant. But yeah, the other genes, similar genes in the locus that appear to be affected by these mechanisms.
Yeah, you had shown with a Manhattan plot with genes being upregulated and downregulated. And one locus which was grayed out was the ApoE locus. So I'm wondering, is there anything which we can still learn from ApoE in the microglia context?
Certainly. I mean, here are sort of all pointed to candidate genes where we know the gene, and we don't have at baseline, so one limitation of this study, all our chromatin QTL, QTL, etc., are derived from myeloid cells that have been unchallenged. They're from healthy or semi-healthy individual, but they're not being challenged with like immune stimulations or other stimulation that may be disease relevant. So we are now writing a sort of a follow-up on this paper using monocytes and macrophages that have been stimulated with either LPS or interferon gamma or oxidized LDL to sort of give them like an overload of cholesterol, which we think is actually one of the likely disease-relevant stimulations. And in that case, ApoE actually comes up. So there may be, independent of the genotype, so there may be some transcription regulatory effects of ApoE or proteins, the ApoE locus, including ApoE, that may be modulating the effect of the ApoE gene. Although, of course, the isoform has such a dramatic effect that those may just be slight modifiers. But we do see this effect in the ApoE region and other regions that don't come up at baseline. Most of the stuff actually appears to be conserved, even with stimulation. There's actually a recent paper that looked at macrophages stimulated with, 12 different cytokines, and also T cells and others, and then looking for enrichment for various diseases. And the enrichment for AD risk in all the macrophages, either at baseline or in stimulated conditions, doesn't change much. So suggesting that there is a core function of macrophages that's being affected, and that's also our thinking here. I mean, P1, of course, is one, and itself is such a major driver, and other more core function macrophages like clearance of apoptotic cells and other debris, to me, are at the top of the list, other than proper immune response stimulation. But we are working on that. Yes, Phil, I know you have raised your hand a couple of times.
Two questions. One, in terms of the reference, if I remember correctly, the Chris Glass data comes from microglia from relatively young people. So how much more, I mean, we don't really know, I think, but how much difference would you expect from older microglia?
Yeah, I guess related to what I was saying, at least for the in vitro system that we have now with macrophages stimulated in vitro, the enrichment doesn't really seem to be dependent on at least the state of the macrophage that we can induce in vitro with those stimulations. I'm also a bit, I'm very agnostic as to at what stage these alleles are impacting microglial function, whether it's the response to overt brain failure that is what you do when you take microglia out of an AD brain, or something, for example, related to myelin homeostasis in mid-age. We have no idea, and I think, of course, it's a good place to start to look at microglia and the concept of NAD brain, but at that point, léger sont faits, everything is already done, and probably this genetic influence has already imparted this risk modulation way, 20, 30 years before the organ has failed, and at that point, microglia may just be reacting to overt brain damage, and they may not be actually really the state that these effects are impacting. Although, from the mouse papers and some of the human papers that are coming out, the DAM, microglia, whatever that is, and you're probably going to be split up in multiple subcategories, appears to have some enrichment, or at least some of the AD risk genes like ApoE appears to be more highly expressed in that state. So that may be a situation where some of these AD risk genes may, impart a stronger effect. But I think a lot of work needs to be done by all of us trying to understand exactly which cells and at what point are doing the risk modulation job. And this may be peripheral cells in 30 years old people, and tough to tackle with drugs, but we don't know yet.
Yeah. And the second question is maybe a little bit more focused. I was looking at your self-one locus with that big shelf of association. I seem to notice that based on the LT, it's possible that there are actually two signals, maybe, two independent signals. So I'm wondering whether you guys, because I don't remember from the paper whether you look for independent effects and whether they both affect the same SP1.
Yeah, if you only look at QTL, it's very difficult to separate SP1 itself from 2 or 3 other genes in MYBC3 especially, although it's a cardiac myosin, it's very lowly expressed in microglia, but it's next to it, and the enhancer activity in that region is so high that I think it's just leaking transcription. But it's difficult because of LD to really tease them apart. So for SP1, we have to do a bunch of other things, but to me, it's really, really interesting, and doesn't come up of the baseline, and that's why we did this oxidized LDL challenge. For me, hoping to highlight or make other association, even within that locus, come up. Because to me, it's really mind-boggling that the 2 transcription factors regulate cholesterol response in myeloid cells, meaning P1 and LXR alpha, is NRNH3, sits next to SP1 there, are in the same locus. And even more surprisingly, SPIB, which is is an ortholog of P1, and LXR beta, completely separate chromosome, they're also next to each other. And we don't know what that means, duplication, evolution, but the fact that they are, and we see this in other loci. It seems that the co-localization of these genes in this loci is there for a reason, for co-regulation or concerted expression regulation, maybe different cell types or different cell states. It's not just random, and they are there together under the influence of perhaps shared regulatory elements, and that's maybe how these regulatory elements impart their disease risk effect by impacting multiple genes, maybe at the same time or at different times for some reason. So.
There we go. It's just a little bit slow. Yep, okay, here we go. Is taking advantage of human genetic diversity to perform basically human genetic screens. And I think as human geneticists, we're very familiar with this type of thinking, but I thought maybe for some of the people, especially who do more mouse work, to sort of think about it this way, because I think you can actually leverage human diversity to do a lot of understanding the regulation of a particular system of interest. So the first vignette is a project that we completed and published a couple of years ago now. And the idea here was to, again, very much in the same vein as what you just heard, but in this particular case, we were interested at the protein level. And of course, it's harder to do large-scale sort of systematic work at the protein level. So we really had a more targeted approach back then, although again, I think this is now getting easier. So again, I think the basic question was very similar. In this case, we had at the time about 23 AD-related genetic variants. And the big question, of course, as we've been struggling with, is how do they interact, or do they interact with one another? Do the different loci influence one another? And so we decided to approach this by measuring the level of expression at the protein level of a number of different proteins, all expressed in myeloid cells. So again, focusing also in the same cell type, myeloid cells, focusing in this case on monocytes. Again, as Edoardo very nicely sort of introduced, there's a lot of shared mechanism between these different myeloid cells. So this is a useful proxy. These are not microglia by any stretch of the imagination, but some of these mechanisms are shared, and I think it's because of the ease in which we can obtain these samples, this is a useful way to explore some of these mechanisms at a large scale. And then, of course, we have to go and validate. So these are some of the genes that we had decided to measure at the protein level. Some of them, again, you recognize well-known, like C33, TREM2, and PTK2B. These are all sort of, we're pretty confident that these are the actual gene that are implicated in the AD susceptibility loci. TREM1 is less well supported. This is sort of work that we had done in investigating in a more focused way the TREM locus. So I would say that's more modest in evidence. TARO-BP, of course, is very interesting, but it came up not because of genetics, but more because of systems biology approaches and so forth. So again, the idea here is the following. So at the bottom, we have the different loci with some of the variants of interest that we wanted to explore. The C33 variant, which is shown here. And we had previously reported that the C33 variant has a cis effect, so it influences the expression level of C33. But the question was, does it also influence the expression of these other proteins that are Alzheimer's disease related? And again, we looked at PTK2B, NME8, and then the TREM locus, which actually hosts multiple different TREM genes. There's 7 family members. And again, in this case, we focused on TREM1 and TREM2, for which we had good antibodies. So in this case, again, nothing very fancy. We basically simply captured data using flow cytometry for each of the antibodies of interest. And actually, I should have focused on this actually here, just to let you know the samples that we picked. So we picked about 150 subjects. Most of them are relatively young individuals who are healthy. This is part of the phenogenetic cohort that is originally I designed and sort of recruited in Boston, but now have transplanted to New York. And they were genotyped as part of the IMVAR project. Actually, this is the panel that you saw from that Edoardo showed, where we had compared T-cell and monocyte EQTLs. So we repurposed the same individuals here. These were all of European ancestry. And then we also used older individuals from the Harvard Aging Brain Study, so older individuals also of European ancestry. In this case, none of these individuals have Alzheimer's. They all are cognitively non-impaired. So again, so the general design, we're trying to find trans-QTL. This is actually trans-PQTL, to be precise, protein QTLs. So we measured the proteins using flow cytometry. And here are the results. So again, the top here, we have the cis-EQTL. So these effects are in cis, meaning that the variant influences the level of expression of the nearby gene. Here is the sort of AD variant for C33. It influences the AD protein in both the discovery sample, the replication sample, and this is sort of the joint analysis. And again, we have previously reported this. It's easy to find. It's a massive effect at the RNA level and protein level. There's a second cis-EQTL independent of the first one in the locus. not as strong, but still substantial. And then here's a TREM1 effect, which I had previously reported, and there's actually a second one over here as well. Actually, sorry, this is the one we had previously reported. There was a new one that we just found in this particular analysis. Interestingly here, and I'm not going to go into great detail here, but again, so these are two SNPs that are independently affecting the level of TREM1 expression. But what was very puzzling, actually, to us initially, was that only this one was associated with amyloid pathology. Again, not genome-wide, but sort of substantial, but not genome-wide significance, evidence of being associated. But while this is the stronger pQTL, this particular SNP was not associated with the pathology, and this one was. So, puzzling. Why would, mechanistically, it sort of didn't seem to make sense a priori. We also looked at PTK2B. This is another actually nice, interesting observation. At the RNA level, the PTK2B cis-QTL is massive, sort of massive effect on RNA expression, barely measurable or capturable in our discovery data set, and then the replication was consistent. So, overall, there's some evidence that this is true. But again, the effect at the protein level in monocytes was much, much less than at the same cell type at the RNA level. So this is nice. These are cis pQTL, again, in some ways not terribly exciting. Really, the more interesting stuff was over here, where we found some evidence for a locus, let's say the C33 locus in this case, influencing the level of expression of TREM2. So this was seen in the discovery samples. We saw evidence of replication, and then this is sort of the joint result over here. This is also true for the NME8 locus influencing PTK2B. Again, replication. For these two, we did not, which also influenced TREM2 and TREM1, we did not have evidence of replication, but this could be sample size issues. Again, these are a relatively small data set at the end of the day. So, so these are sort of the two trans protein QTL that we reported in this paper. And so this is just to illustrate, what we saw. Here are the genotypes for the C33 variant, C being the risk allele in this case, and here's the level of expression of TREM2 on the cell surface of monocytes. You can see a nice dose-dependent effect with the risk allele having higher level of TREM2 expression than the protective allele homozygotes over here. Now, the interesting thing is what we also did is to try to validate this. We took an antibody, an anti-C33 antibody, to engage C33. And what happens is C33 gets downregulated from the cell surface when this happens. And if we measure TREM2, the immunofluorescence intensity of TREM2 on these cells in the context of anti-C33 or an irrelevant isotype control, you can see that these are all individual subjects. I forget how many there are, maybe about 15 or 20 individuals. All of them decrease after we expose them to anti-CD33, suggesting that so again, there's a relationship between C33 SNP variant and TREM2 expression. And if we engage C33 and this decreases in the cell surface, then for a mechanism that we don't know today, this influences TREM2 expression. And this just shows that TREM2 expression goes up with amyloid pathology. Which is the pathology with which C33 was associated. So again, this is one way in which we were able to connect, two different loci, which are not on the same chromosome, C33 and TREM2, and to begin to understand, to assemble these protein level networks. Interestingly, if you did the same analysis at the RNA level, you didn't see anything. There was no connection of C33 on TREM2 expression at the RNA level. So this is something that's happening at the protein level, mechanism yet to be determined. So this is sort of summarizes what we found in this particular work, where again the C33 variant somehow influences TREM2. Now going back to the TREM1 story, if you remember, again there were two cis-QTLs, they were both cis-pQTLs. This one was the weaker EQTL and pQTL. And yet, this one was associated with disease, and the other one, which was a stronger pQTL, was not. The reason was that actually this particular variant, the other variant influenced both TREM1 and TREM2 expression in the same direction. So the ratio of TREM1 to TREM2 doesn't change. But this one only influenced TREM1, and so as a result, there was a difference in the ratio of the two receptors. And all these receptors, of course, they don't work in isolation. The cell surface is constantly sampling the environment, and these different receptors are interacting and signaling, at the same time, and these signals get integrated. So again, showing that the relative presence of these different receptors probably is important, in the context of AD. And here's just the NME8 case where, again, by a mechanism we couldn't measure at the protein level, but we found an effect of NME8 on PTK2B in this case. So again, that was published a couple of years ago. And then my biostatistician, Charles, decided to do, so this is great. Of course, initially we had been very focused and just to do a very targeted transit association, because we know that we have very little power to do this type of analysis in general. But, Charles said, well, what the hell? Let's take a look and do it genome-wide. So he did. And so in this case, here we're showing 4 genome-wide association studies. And then each of these colors represents basically one GWAS for different proteins. And so it's labeled here. This is TyroBP. This one is C33. So if you look at the C33 level of expression, this is a genome-wide result. This red circle here is the threshold of significance, genome-wide, correcting for 4 GWAS. And you can see the very strong peak over here, and this is the C33 locus. So again, this is a cis pQTL effect that we expect. Similarly, for TREM1, that's shown over here. Here's the red results are for TREM1 expression at the protein level, and we can see again the TREM1 locus has a very strong effect. Now, the surprise was actually that we found 2 more genome-wide peaks, one for TyroBP and one for TREM2. And in both cases, it was the same locus, actually, SIRPB1. And this was done on 226 subjects, the same subjects that we had used for our targeted study. And here are the sort of the local, local zoom plots. Results for TyroBP here, results for TREM2. And what you can immediately appreciate, I think even just visually, is that the 2 peaks of association are morphologically different. And that's because they're actually 2 different haplotypes. And we formally have shown this, that this tag SNP and this tag SNP are tagging different haplotypes within the SERP locus. And this, like TREM2, SERP, the SERP family members are all located together. There are multiple ones in this particular area. So again, but still very interesting. So we have in one locus, and the variants are located basically in the vicinity of the SERPB1 gene, but formally it could be other genes being affected as well. But again, 2 different haplotypes, but the same locus. Then what we tried to show, and this is actually just showing the result in a different way. Here are the genotypes for one of the 2 variants, the one that influences TyroBP. You can see again a nice dose-dependent effect. Same thing here for the other locus. This is for the other variant. This is the TREM2 expression. You can see again a nice effect here. Now, if you look at this particular variant that influences TyroBP, this is pretty clearly a cis EQTL and PQTL and PQTL for SIRPB1. So here we have SIRPB1 RNA expression. So this is the same variant as over here, the same genotypes. You can see again a nice dose-dependent effect. And here's the protein level data, again, very consistent. So again, presumably the mechanism in this case is that this variant influences SIRPB1. And I'm not going into this today, but we looked at multiple other SERP genes. This is, although we can't be definitive that there's no other SERP gene that's affected, this is the one that's the most likely candidate so far. We went on to look, so these are in monocytes. That's nice, but can we actually go a little bit further and show that in the context of microglia-like cells, that this is also true? Again, it would be nice to get microglia EQTL or PQTLs. Those are not available yet. Again, I think a lot of us are working on this, but it will take a few more years probably to get good results. In this case, we use microglia-like cells that are derived from monocytes. And here again are the genotypes of this particular SNP. And you can see that the effect is preserved in these cells that are, again, derived from monocytes but have many microglial features. Separately, this is the other variant that influences TREM2, and we had a different data set that we had generated on the monocytes from individuals with Parkinson's disease, and we basically were able to replicate the effect of the SIRPB1 SNP on TREM2 expression. In this case, actually, it's a mirror image. It's flipped from the other one, but the result is consistent in this data set. We were able also to show, as had been shown previously, that SERPIB1 is expressed in microglia, and that it's actually found in the vicinity of amyloid plaques. So here is a microglia that's sort of labeled with, so it's a little bit IBA1 in green and SERPIB1 in red. You can see this particular one is yellow, so it has both stains, and here it's in the vicinity of the amyloid plaque over here. Also, so that's great. So there's an effect on protein level expression. Does this variant have a functional phenotype? So yes. So again, looking at the monocyte-derived microglia-like cells, here is the variant that influences TYRO-BP, and we can see that the allele that has more SERPIB1 and more TYRO-BP has better phagocytosis, so better uptake of A beta in that case. And this is just shown here. You can see this is the uptake of individuals who are homozygous for the G allele versus an individual who is homozygous for the T allele. And each dot here is one individual. Finally, we tried to validate this in a different way by knocking down SERPIB1 in this MDMI system using shRNAs. And so what we did here is we knock it down, and so first we look for expression of SERPIB1 on the cell surface. This is the target, of course, of the shRNA. So nicely, you can see that I think there are 10 individuals here. In these 10 individual cells from 10 different individuals, we see a consistent knockdown of the protein level of SERPIB1 with the shRNA against SERPIB1. You also see in the same cell, if you measure TREM2, a decrease in the TREM2 level expression, a decrease in TYRO-BP, and diminished uptake as well. So again, I think we've now implicated SERPIB1, or at least the SERP locus, certainly, as a regulator of both TREM1 and TREM2 and TYRO-BP, two of the important genes that are involved in AD pathology. The other interesting thing, actually, is that SERPIB1 emerged as part of the ADSP as one of the candidate loci that harbors rare variants. It failed to validate in the original, the validation has been done so far, but it's still underpowered, and so we'll see what will happen with that. But there certainly are, at least some excess of variants, AD variants, in this particular locus. Finally, I'll show you a third vignette. Let me see what time it is. Yeah, so we have 5 more minutes. This is something actually I've presented here before, so but I think this audience is a little bit different, so I thought I would show it also here. Again, the 2 other examples are leveraging peripheral blood cells as a model for the cells that we care about. Although again, formally we don't know whether the monocytes or the microglia are the most important one for AD. In this case, we want to understand drivers or sort of regulators of microglial activation. And so to do this, we repurposed a data set that our colleagues at Rush University had generated, where they had actually measured that counted the number of microglia of different morphological types. So stage 1 are the ones that are nicely ramified, and they're thought to be the ones that are mostly in a homeostatic state. And then as the cells become activated, they adopt the stage 3 morphology, which is much more rounded with fewer processes. And so our colleagues had counted the number of cells in stage 1, 2, or 3 in 225 individuals in 4 different brain regions, cortical and subcortical regions. We then compared the presence of different subsets of these cells in the cortex or subcortical regions. I'm not going to talk about the subcortical region because basically it was all negative. So focus on the cortex, where both in the inferior temporal lobe and the midfrontal cortex, we see the same consistent result. And that's that if you look at the total number of microglia, so all microglia stage 1, 2, and 3 together, we see no difference in the total count whether individuals have AD in green or don't have AD in red. Now, if you look at only the stage 3, this is where there is some difference. And you can see that individuals who have AD, there's a shift to the right, so there's a great proportion of these activated stage 3 cells at that point. And then we can refine this a little bit further by calculating the proportion of the activated microglia, so the stage 3 divided by the total number of microglia measured. We could then sort of take a look at this trait a little bit further and see, okay, well, what does it relate to? Well, it relates to amyloid and tau pathology. So basically what we're showing here, this is a threshold of significance, is this red line here. The darker red line is, or the darker dots are the inferior temporal cortex result. The midfrontal are in yellow. Again, blue and purple we'll ignore. Those are the subcortical regions. The associations are with amyloid and tau measures. If you look at vascular measures, TDP-43, the Lewy bodies down here, there is really no association, at least in this particular regions that we looked at. And with cognitive measures, again, there's a consistent effect of the cortex on multiple different cognitive measures. We were able to do some modeling, try to infer the where these activated microglia may be having a role. Do they come before amyloid, which then leads to tau? Does amyloid come first, and sort of they're playing sort of a mediator role to cause tau pathology, et cetera? So we considered a number of different models, and then this is the most parsimonious model from these particular data. Again, this is not causal in that this is just a model of what a hypothesis based on our cross-sectional data. But basically, amyloid pathology and activated microglia independently have an influence on the accumulation of tau pathology, which then causes cognitive decline over here. So here we're able to sequence exactly where these cells may be playing a role. And again, perhaps they may be important in influencing the deposition of tau pathology. Well, we only had 225 subjects. Dan, my postdoc at the time, who now has his own lab in Toronto, decided to do a GWAS, and remarkably, actually one locus was significant at the genome-wide level. This is shown over here. And this is sort of the effect on the trait. So here's the allele that is associated with more, with a propensity to have more activated microglia in your brain. It's when you're older. And we didn't have a second data set to replicate this, but we looked actually at a different set of individuals who had PET data for microglia, and we saw a similar effect on the PET data for this particular locus. And then we did some polygenic modeling to see, okay, so if we take a look at all the entire genome, and there's some enrichment for associations with the trait of interest, what does this genetic architecture relate to, or where's there overlap with this genetic architecture with some disease or other traits? And the strongest overlap was with Alzheimer's disease, both for the inferior temporal cortex and the midfrontal cortex over here. Again, what was quite interesting and a little intriguing is that educational attainment is the second most sort of enriched trait. But again, sort of this fulfills our expectation, and I think refines the finding that it may not be just all microglia that are involved, but really these activated microglia genetically may be the targets for at least some of these AD variants. And this is just a different way of showing the same thing. So again, so in terms of this part, we were able to sort of draw this hypothesis that this proportional activated microglia is important for tau accumulation. But in terms of the genetics, which is really the focus of today's talk, again, it's just an illustration that by leveraging the diversity of human subjects and by capturing a phenotype in a large number of individuals, we can do these types of genetic analyses that are really essentially genetic screens looking for loci that control this particular trait. I didn't really get into it, but the locus that we found here, fortunately, there's no obvious gene that may be the target here. It's a little bit of a gene desert. There's some non-coding RNAs, but there's nothing really obvious. But again, so we're trying now to expand this data set. And with this, I'll close and highlight a few people. I think, unfortunately, Dan may no longer be here because he now has his own lab in Toronto, as I mentioned. He did the microglial story. Kruti Patel did the nice work on the SIRPV1, working with Charles White, who did the original analysis. And that's a collaboration with Betsy for both the original sort of targeted work and with the follow-up that we did on the genome-wide level. Thank you.
So the correlation you were seeing between the SPAM and educational attainment, is that positive or negative? If I have higher educational attainment, am I more likely to have Alzheimer's disease, or presumably less likely?
It's just sort of an enrichment, for association. So we didn't look at the directionality in this
Okay.
case. You can't infer it from that graph.
Okay. From the graph you can't, but from the data you could, right?
Right.
Because
Presumably, yes. I think presumably the more microglia you have that are activated, the more higher the level of cognitive attainment on average. But I'd have to go back and double check. And whether this has to do with, we can speculate that this may have to do with sculpting of synapses during development and later on.
Yes, we actually done neuromodulation, educational attainment, AD, they share genetics, and so, it's non-effect, not on microglia, but on the two traits. The higher the educational attainment, the least risk. Yeah. So anyway, for the PQTL studies, have you confirmed that these are actually PQTLs? Because with like some all IZ or something, or are these, because if these are only surface, are we just looking at receptor internalization QTLs? Because many of these are receptors that may be in the same, CD33 and TREM2, ITAM, ATIM, PTK2B, ThyroBP, these are all part of the same signalosome probably. So can we, maybe it's just internalization of the receptor from the surface.
You're saying, is your concern that the receptor is getting internalized while you're staining it?
That the effect that you're seeing on cell surface expression is not a protein QTL per se. It's not the total level of protein that's being changed, just the level on the surface that's changing. Hence, it's an internalization QTL rather than an actual protein QTL.
Although I think, again, with staining is done at 4 degrees typically, so I don't think there would be much reaction to the...
No, but before the staining, I'm talking about...
So I think we don't go into... with this approach, we couldn't differentiate exactly what the mechanism is, right? All we can say is that based on what we've measured on the cell surface, this particular locus influences that particular trait.
Okay.
How it happens, it's a black box, that we have to investigate this further. But, the important thing, the first step was just sort of linking these different loci to one another, and then to begin to explore, I think what we've done since then is to begin to assemble protein networks by doing co-IPs to identify the binding partners. But that's, it's long road.
So you've nicely shown the regulation of TREM2 and ThyroBP by SIRBP1, right? If you take enough patients that carry mutations that impair the regulation of those 3 genes, do you see anything that links the severity of the disease or the age of onset or anything?
Right.
And so that the imbalances in any of the 3 could actually lead to the same outcomes, right?
Right. So far, the only one that has a robust genetic association to Alzheimer-related traits is TREM2 itself. So that's the original. ThyroBP, again, has no, as far as I know, that has no variant that relates to AD directly. It has an influence on some other diseases that are, that are sort of that are related, but not AD. In terms of SIRPB1, again, there we looked pretty hard. We didn't find any. The variants that I mentioned don't seem to influence the traits that we've measured, like amyloid and tau and cognitive decline. So there's no association there, or syndromic diagnosis of AD in the data set that we look at. But it's limited in size, about a couple thousand people. That said, there is that one observation that in the ADSP, the sequencing project, SIRPB1 is enriched in the discovery study for rare variants. So maybe this has to do. So there's some hint of it, but it has to be yet to be validated.
I was interested in the relationship between these stage 3 microglia and cognitive function and the relationship to pathology. And I was wondering if you or any of your colleagues at Rush had looked at microglia morphology in resilient individuals that have pathology but are still cognitively intact, and how the microglia look in that situation.
So it's a good question. We have a trait that we call residual cognition that sort of captures that. It's basically how well you're functioning based on the amount of pathology you have. It was not on that particular figure, and I don't remember if we looked at it. And at the end of the day here, we only have 225 people. It's a large, substantial number, but we want now to do the entire collection. But this microglial staging is actually quite onerous and challenging. So it's been a little slower than we hoped to generate more data. So maybe one last question while we switch, because I think we should keep on time. So one more question.
Yeah, I have a question regarding the EQTLs for monocytes, which you had found, or QTLs, which you had found for the AD risk genes. Do these also affect monocyte counts in the peripheral blood, and would we expect this to be the case? And if so, in which direction?
That's a good question. I think we may have looked at this at some point, but I don't know offhand. We'd have to double check. It's possible that it does, but again, the association is with these activated morphology, and that would probably happen in the tissue, because the monocytes are relatively naive cells. They don't differentiate and adopt their final differentiated form until they get into the tissue. So I would expect if you characterized macrophage in other tissues, this variant might have an effect there. I mean, I might expect that. But I'm not sure if we would see it necessarily in the monocytes, which are relatively naive cells. So now we have our last and final speaker, our illustrious colleague, Hemali Phatnani, who is going to talk to us about the Center for Genomics of Neurodegenerative Disease.
I'm going to try and keep this as brief as I can because I'm the only thing standing between us and beer. So ALS genetics, so the Center for Genomics of Neurodegenerative Disease, which I direct at NYGC, essentially has three main goals: to serve as a hub for collaborative interactions, to build tools and resources that we can disseminate to the community, and to apply genomics to understand the molecular basis of disease. And I'll tell you about that in the context of ALS and FTD. But we aim to integrate studies across multiple scales and to integrate multiple data types across these scales. So in the context of the ALS-FTD spectrum, we're looking at how can we get enough ALS patients to one day enable the kinds of studies that you just heard about in AD. So we take well-annotated patient samples, do whole genome sequencing from tissues from these patients. We also do RNA sequencing to look at the relationship between DNA and how it's expressed in the central nervous system. In parallel, we do studies on animal and cell-based models to interrogate in specific cell types how gene expression changes in time and space. And I'll talk about this half first before I go into how we're doing the interrogation in time and space and integrating different data types. So ALS is a relatively rare disease. There are about 25,000 patients with ALS in the US today. And so you're faced with two types of challenges. The rarity makes it difficult to obtain large numbers of samples that are clinically well annotated. It's also clinically heterogeneous. So if you want to, which means that it presents differently in different individuals. It can set in at different places, proceed at different rates and patterns, may or may not be associated with cognitive deficits that may or may not reach the threshold for frank frontotemporal dementia. So if you want to understand the relationship between the genotype and how these effects present themselves in individuals, you need to have very well clinically annotated samples. Because it's rare, it's really difficult to get enough samples for these kinds of studies from any one clinic. Even the busiest clinic in New York is going to see maybe 400 ALS patients a year. So you have to basically pool samples from a number of different places in a way that they can be studied together. And the clinical heterogeneity makes that difficult. The other challenge is one that the entire field of neurodegenerative disease faces, which is how do you obtain mechanistic insights into how mutations actually cause disease. So the solutions that we've envisioned to overcome these challenges or address these challenges is to obtain samples. We established a federated program with centralized governance. And I'll tell you what that means in a bit. To obtain mechanistic insights into the disease, we have our own small in-house program, but we also partner with labs all over the world to be able to make the best use of the data sets and the resources that we generate. So here are some of the federated partnerships that we have at NYGC. There are multiple sites all across the United States that pool their data and for whole genome sequencing here at NYGC. These represent about 32 sites in 6 different countries all over the world. So the centralized governance of such a highly federated program essentially involves us being a coordinating center where we develop and disseminate SOPs for how the data ought to be collected, how the samples ought to be treated, what clinical data we need, how can that be standardized and harmonized, how do we warehouse that data to make it analyzable together and shareable, because all of that requires a lot of regulatory paperwork as well. So to date, we've sequenced almost 4,000 DNA samples and about 3,000 RNA, 2,500 RNA samples. This is now the largest ALS whole genome sequencing study in the United States. Now, we said that we want to make this a resource that can be mined by people anywhere. So data sharing is a huge component of the work that we do. And we distribute the data from these sequencing efforts all over the country and all over the world. And you can see there are more red pins that get the data than there are red pins that give the data. And that's because we want to decrease the time it takes between generation of data and analysis of data. So if you've got a new pipeline or a new way to look at the genomic data that we're generating, you just have to request it from us and we'll share it with you. The advantage of an approach such as this is it decreases the time to discovery, and the same data set can be analyzed in multiple different ways. So to date, that data set has resulted in the identification, combined with lots of other things, of 5 additional ALS-associated genes. I'm going to skip this, as has the RNA data. So now I'm going to get to the integrated analysis across scales that I talked about, which is the right-hand side of this figure. So this bit and this bit are going to meet in the middle here. And we said we want to look at how genes change their expression over time and space. And why do we want to study spatially resolved transcription in ALS? Because like other neurodegenerative diseases, it involves pathologies of multiple cell types. And we want to understand how these different types of cells interact as disease progresses, what the dynamics of the ALS pathology are across time and space, where does it start, and how does it spread. To understand spatiotemporal dynamics like this in all relevant cell types at the same time means that you need a way to measure transcriptome-wide gene expression in a way that preserves spatial information. And these observations need to span disease progression. So how do we do this? How do we measure gene expression in multiple cell types and locations simultaneously? We do it by using a technology developed in Joachim Lundberg's group in Sweden called spatial transcriptomics, or spatially encoded RNA-seq, where essentially you take a glass slide that has a printed pattern of oligo-DT probes that have unique identifiers that identify where on the glass slide each probe is and allow you to count how many species of an individual molecule you captured at that spot. You then take a section of fresh frozen tissue, lay it on the glass slide, stain it with H&E, take a picture, permeabilize the tissue, let the RNA diffuse and be captured by these oligo-DT probes on the slide. Do your library prep like you would any other library prep. Remove the tissue, cleave the library off the glass slide, and sequence as you would for any RNA-seq experiment. The advantages of this approach are that it's high throughput. You don't need to select genes ahead of time that you want to probe. The workflow is really simple, can be done in any lab. You don't need special equipment. It's relatively inexpensive, but not at the scale at which we did it. But you need to do it at that scale to know at what scale to actually do it. And it can be used on nearly any sample type. The cons are that it has moderate spatial resolution, and as it stands, it has no cell type specificity. So the probes that I talked about in the iteration of the technology that we used was spotted in 100 micron circles with a center to center distance of 200 microns. Now, as you lay the tissue down, this can span anywhere from 2 to 20 cells. I'll show you a conceptual video of how the technology works before I tell you how we designed the experiment and analyzed the data so that we could circumvent some of these disadvantages. So this is basically how the technology works. You can apply it to any tissue type in any species. You take a piece of tissue, you slice it up into really thin slices. It's frozen tissue, although people have tried it in fixed as well. Doesn't work nearly as well. This tissue is then laid onto that glass slide that has this array of probes spotted on it. You're going to take a picture after H&E stain. You then permeabilize the tissue so that the RNA diffuses out and is captured by the probes spotted on the slide. You do your synthesis, first strand, on the slide. Then you cleave that off the slide, remove the tissue, and then sequence as you would in any other experiment. I know you know how sequencing works. I just like how the letters light up. So then you put that back in the computer and you map, because you know where each spot came from, you can map the expression back onto where it came from on the slice and stitch them all back together to give you a 3D map of what was expressed where in the tissue. Now this doesn't allow you to do any differential expression. You're just looking at this piece of tissue in this video. I'll talk you through how we did that. But first we did the proof of concept experiment where we took a slice of fresh frozen spinal cord from a mouse. That's the H&E image on the left, and a cDNA print of that piece of tissue on the right. And here it's a lawn of oligo-DT. We had to work out basically what the permeabilization conditions were going to have to be in order for us to get this experiment to work on spinal cord tissue. So we're just incorporating Psi3 on a lawn of oligo-DT, and you can see that this is a pretty faithful representation of this. And because you can map this to this, you can also measure how much lateral diffusion there is. It's not much, 2 to 4 microns laterally. So now I said that you don't get differential expression the way the setup was originally devised. So we had to overcome a lot of computational challenges associated with the spatial transcriptomics methodology and develop a whole new framework for data analysis. And that was done by Rich's group, who's sitting here in the second row. And here are the kinds of challenges that are associated with data sets of this type. It's very high dimensional. It's across the transcriptome. There are 1,200 tissue sections from both mice and human, multiple animals, multiple time points, multiple genotypes, multiple regions across the cross-section of the cord that you need to factor in. There's also highly variable zero inflation, which means that you don't know whether a zero measurement is actually zero because there's nothing there to be captured, or because your sequencing depth was too low to be able to capture it even if it was there. There's also a lot of variability between spots and between samples. So Rich's group came up with a really sophisticated way to be able to factor all of these different things into the analysis. And they built this hierarchical generative probabilistic model, which you can ask him about over beer, because I'm not qualified to discuss it. But essentially, in order to be able to borrow information from neighboring spots, which is what this model allows for, we had to first group the spots in a way that made sense. So we divided the cross-section of the spinal cord into 11 different anatomical annotation regions based on whether they were in white or gray matter, medial or lateral, dorsal or ventral. And then we took each of the SD spots and assigned them to one of these 11 AARs, or anatomical annotation regions. And there were, about 150,000 spots that the team manually had to assign to one of these different AARs. Now, Rich's group also developed an automated way of doing this, so none of you ever have to go through that pain. What this allowed us to do is to be able to register the different slices, one on top of the other. And through extensive replication and this kind of registration, just using the centroids of the dorsal and ventral horns, we were able to overcome the relatively coarse sampling afforded by the design of the array and achieve pretty dense and even sampling across the cross-section of the cord. So these show you 4 different slices of mouse spinal cord cross-sections on an SD array, which is about 6 by 6 millimeters. And you can see that the spots here, because they're 100 microns with a 200 micron center-to-center distance, are pretty sparse. But this is much, much denser than that. So then this is the information that gets fed into this model that accounts for where the spot came from, which animal it came from, where on the spinal cord it was, what the genotype of the animal was, etc. And it allows you to quantify within each region how the measurement of, before I go into that, it also, because of this model, it also allows you to infer the expression of many more genes than you can actually capture directly. So we can capture maybe 2,000 genes, the expression of maybe 2,000 genes, but we can infer the expression of about 14,000. And it allows you to quantify the difference in expression within each anatomical region between the control and your experimental system. So here's what that kind of quantification looks like. For example, here's GFAP, which is present in reactive astrocytes that you would expect to see in this kind of system. And here's a ribosomal protein that you don't expect to see changed. So this is for each region, each anatomical annotation region. And these 4 rows are for the different time points. The width of each, the width here is indicative of how confident we are in the measurements, and the distance between the two tells you how different the expression is. So you can see for GFAP when the wild type and the mutant start to differ. Oh, and the blue is wild type and the red is mutant. You can see when the wild type and the mutant start to differ, where that happens, and how this change is propagated over time. So we did all of these experiments first in a mouse model of the disease to build all of these workflows and the computational methods, and then we applied the tools and the techniques that we developed to post-mortem human samples. So now we've inferred the expression of about 14,000 genes. We can quantify the difference between them, between wild-type and mutant animals over the course of disease. So then we grouped them based on how their expression co-varied in all of the different spots. And based on this grouping, we came up with these modules of co-expressed genes. When you take each of these modules and overlay them back onto your ST coordinates, you can see that these gene expression modules correspond to distinct spatiotemporal domains. They have, each of these modules has distinct spatiotemporal expression profiles. So module 1, this is the wild type, this is the mutant, is very different from module 11, for example. And each of these is very distinct from these two. So this still doesn't tell you, so this tells you where and how this set of genes is changing over time. It doesn't tell you in which cell types this is happening. To do that, we actually deconvolved these data using independently generated single cell or single nucleus sequencing data. So we said how much of this expression can be explained by expression in distinct cell types. So you can see that by doing that, we can take module 8 and separate it out into what's the contribution of microglial genes, what's the contribution of astrocyte genes. We can do this for each module. Because we can do this for each module, and each module has a distinct spatiotemporal expression profile, what we can essentially infer is how subpopulations of specific cell types are behaving across disease. So here we're saying that there's a submodule 17 in module 8 that represents microglial genes. We also did the same experiment in a mouse where we had knocked out the expression of ATG7, which is important for autophagy. We knocked it out in cholinergic cells and looked at how, in the SOD background, and looked at how expression was affected for all of the things that we measured. So we know that module 17 is a microglial enriched module, a submodule of module 8 that's affected by perturbation of autophagy in motor neurons. And this is another way of looking at this data. Similarly, microglial module, submodule 12 in module 1 has a very different profile. And this is IBA1 and this is SAL1. So IBA1 and SAL1 represent distinct microglial subpopulations that behave very differently in disease in terms of how they evolve over space and time and are differentially affected by modulating autophagy in motor neurons. We can do the same thing for astrocytes. GFAP and SLC7A10 identify two distinct astrocytic subpopulations. We can also parse out the activity of specific signaling pathways within each of these modules and confirm this at the level of microscopy. So here we're looking at ThyroBP and TREM2 at how their expression changes over time and space. So ThyroBP and TREM2 essentially are acting here. Is this, can everybody see this? Yeah, this is, Salas always has two different versions of this in case the projector is crap. So here we're looking at now immunofluorescence images in a wild type and a mutant animal for motor neurons and ThyroBP. And this is the ST data that's telling you that ThyroBP expression is upregulated in the ventral horn and the ventrolateral white matter. And this is confirmed by the immunofluorescence staining. And this is very, very early in disease, long before you see symptom onset. If you look really up close, you can see that this is happening really adjacent to the axonal tracts of the motor neurons. So we applied this system and these tools that we had developed to the analysis of post-mortem human samples. And we did this by stratifying the patient samples based on site of onset. So we looked at patients that had bulbar onset ALS, and we looked at patients that had limb onset ALS. And we looked at the cervical spinal cord and the lumbar spinal cord from each group of patients. ALS is thought to be focal in onset and then spread through contiguous regions. So if you look at the lumbar cord from bulbar onset patients, it'll be relatively better preserved, less affected in disease than lumbar spinal cord from lumbar onset patients. So by looking at how gene expression varies, the further you are from site of onset, we can start to see very similar patterns of expression, spatiotemporal dynamics of gene expression changes over the course of disease from end-stage postmortem samples. And a lot of the mouse modules that we find are also replicated in human samples. Now, what we know from doing this study on 7 patients is that we are underpowered to actually do quantitative differential expression measurements. So we are now engaged in expanding the size and scope of the human study to be able to make similar kinds of differential expression measurements in human samples as we did in the mice. All of these data are available for you to view and query at this website. You can look at the array level data for mouse and human. You can look at digital in situs for individual genes or groups of genes in mouse and human. You can look at the differential expression view. You can look at the results of the differential expression analysis. You can also look at the trajectories of groups of genes and how they vary over time in different regions that we annotated. So to summarize, we used the mouse model to build a robust workflow that allowed us to apply spatial transcriptomics at scale. We developed novel computational approaches to analyze these kinds of data that allowed us to identify spatiotemporal dynamics of gene expression programs. We could identify regional subpopulations of glial cell types that we confirmed using a microscopy. We know we can apply ST to study the human spinal cord. We now also know we can study human brain using this. We built an interactive portal for open access to ST data. So in the next steps, as some additional approaches to overcome the lack of single cell resolution here, we want to fuse this ST modality with other modes of looking at the same data, the same tissue section. So we want to look at how gene expression changes in regions that are close to regions that show neuropathological disease burden, like TDP-43 aggregates, for example. We want to combine this ST workflow with imaging mass spec workflows. We know that we can do this at the wet lab level, and we need to develop computational tools to be able to merge these two different data types. And finally, we want to integrate this, well, last but one, we want to be able to integrate this with multiplexed immunos. So we've adapted a tool called Indirect Iterative Immunofluorescence imaging that allows you to look at, allows you to do IFs essentially on the same tissue section by a series of binding and elution steps in a buffer that prevents cross-linking of the antibody to the antigen when you image. We know that we can do this iteratively for up to 8 cycles. That means that we can look in the same tissue section at single cell resolution at the protein level and interrogate up to 24 separate antibodies. We can also corroborate this using in situs. So we've developed imaging transcriptomics measures that can integrate with the ST measurements using StarMap. And our focus is on expanding these to be able to integrate these different data types so that we can overcome the loss of single-cell resolution in ST data in postmortem human samples. So I'd like to end by acknowledging everybody who contributed to this effort and all the people who paid for it. This was across 3 different groups at NYGC and at CUMC, at the SciLife Lab in Sweden, and Rich Bono's group at NYU and the Flatiron Institute. And I'd like to thank you for sitting through 3 talks, so now we can go get beer.
Hi, thanks. I may have missed it. What's the temporal resolution?
It's a snapshot.
Yeah, but what is the like in what units?
We took it every month.
Every month. Okay. And also, I understand how you can correlate spatial positions between slices of the same sample, but how can you, other than mapping them to anatomical regions, can you actually correlate spatially, quantifiably, the distance between spots across samples, like in the human brain, where it's notoriously difficult to orient and align?
You mean on a particular slice?
No, like any point, any cell or any region in one sample. Like how close is it to another one, and like can it actually help you define the tissue or cell specificity, or is the resolution just all these points map to the same anatomical region between patients?
The anatomical regions are what we defined.
Yeah.
Using features that were anatomical features of the tissue itself. So if you're looking in the cortex, if you look at where you are, which layer you are in, and you have markers for that layer, you should be able to do exactly the same thing. You can also look at the spot level, which is another thing that we're developing, and based on the nature of expression that you see within a particular spot, you can start grouping the spots as being more or less similar to one another. So you can assign a spot to a particular region based on features of that region, or you can go from the spot level and start building up relationships that way.
I was interested in how you were able to expand from 2,000 measured genes to 14,000 inferred genes. And is that relying on the couple times they are measured, or are you pulling in different data sets, or how does that work?
I'm going to let Rich take that question because it involves going into details of how we're borrowing information.
Sorry if that's too technical.
No, it's so there's on average 2,000 measurements of different genes per spot. But then across the, all of the data sets, all the different spots, all the different slices, we see all those 14,000 genes many, many times. And so essentially you might see a spot that has, this gene, and then you might see a spot in the same type of mouse, all the various other metaparameters are the same. It's in the same region. And so what the model's doing is it's building up an estimate for the region. But for each of those sort of points in the common coordinate, there's many, tens or hundreds of spots supporting it. So it's really through this aggregation of all the data into the common coordinate that you see all those things. But yeah, everything that's inferred, there's many, many measurements of that gene before we make the inference.
So is this what appeared to be like an early reactivity of microglia in white matter? Something that you unexpected or known to pathologists for 200 years? I'm no ALS expert, so I have no idea. And more importantly, how does the expression of disease genes map to that?
We haven't done the mapping of the disease genes to that. There is no TREM2 or TYROBP are not implicated as ALS-associated genes. We were not expecting to see this in this region at this time with such specificity. So it's something that we are keenly interested in following up on. very much.